I'm Tim Ventura, and we're joined today by Liz Parrish, MBA, the founder and CEO of BioViva Sciences USA. Liz made medical history and international headlines in 2015 when she volunteered to be the test subject for an experimental procedure that lengthened the telomeres in her cells, which might just be the first step towards future treatments to reverse aging. Liz, welcome. Let me start out by asking what inspired you into the field of anti-aging medicine, and how did that lead you to found BioViva?
Thanks, Tim. Well, number one, I'm happy to be here. And number two, let's get right into it. So in 2011 to 2013, I volunteered for a project that was based around the education of the use of stem cells. And stem cells were a really exciting technology. They still are. But I fell in love with what was happening at the genetic level of the stem cell. So all the cells in our body have the same genes. Some of them look like our nose and some of them look like our toes, but the stem cell has the ability to regenerate tissues and heal us when we get injured. And that was really fascinating. And I didn't know how important that genetic journey would be until 2013, when my son was diagnosed with type 1 diabetes. So here I am, I'm in the hospital. I've spent 2 years learning about regenerative medicine and genetics. And I asked what was available for my son in order to cure his type 1 diabetes. And I was just told that, I should be happy that he has a treatable disease, that kids there are dying of diseases, and that stuff was considered experimental medicine and not used in children. And it kind of shocked me that people would talk about experimental medicine as being such a negative if kids were dying. And it just didn't fit into my head that we allow people to die because we're afraid of some medicine when we know that all medicine is an experiment. And so anyway, I went looking for cures for kids, and I found a conference that was speaking on genetics. It was an aging-based conference in which I didn't know what the relation would be, but the professor of genetics, George Church, was going to be there, professor at Harvard, that is. And I was excited to meet these people and see what type of treatments could translate to children. When I was there, I kind of fell in love with aging. It was explained to me that, doing experimental work in children was considered very bioethically uncouth. But there were millions of people that died every year of aging diseases. And a lot of the technology that they were talking about that would treat biological aging would also treat a childhood disease. And so anyone, who looks at our company and what we do now, all of the genes that we choose would treat also a childhood disease. And so they have the regenerative capacity on one side for an aging population, and then benefits to specific diseases that children might have. So by treating an aging population, we can expedite the therapeutics to children. And that was really, really exciting to me.
Well, so to help our audience understand better what you're doing, let me ask now if you can give me kind of a high-level explanation of why telomeres are important to aging and how your company was able to lengthen them for you.
Right. So telomeres are the caps at the ends of your chromosomes. They protect the chromosome. They basically create genomic stability. Genomic instability on the opposite side leads to things like cancer. Okay, so that's why you would want your genome to be very protected. They also help protect the mitochondria. So in research that we did and research that other groups have done, we've shown that mitochondria are healthier, more stable, and larger in cells that have been treated with telomerase reverse transcriptase, which is the gene that lengthens telomeres. It creates the enzyme telomerase. So this is a really important treatment because one of the hallmarks of aging is telomere attrition, but other hallmarks of aging are benefited from lengthening telomeres. So the hallmarks of aging are the things that are happening at the cellular level. They're why your cells are degenerating, and they essentially the shortening of these telomeres are associated with all of the diseases of aging. So as our telomeres get shorter, we get more genomic instability, our mitochondria dysfunction comes into play, our intra and extracellular communication starts falling apart. And so we get things like stem cell depletion, and that means we have less regenerative capacity. So lengthening your telomeres is essential. So it's essential to extending healthy lifespan. Let's make it really clear. Lengthening your telomeres may not cure all hallmarks of aging, but they are essential to your body, your organism living longer, healthier. So if you don't deal with the telomere problem, even if you reset the cell's epigenetic age, you still will hit a cellular division limit. So there's a myriad of species. There's about 25 species now, and it's growing, that we know it's called a Hayflick limit, based on the length of the telomeres and the rate of shortening. So some species, like there's a sea urchin that lives really long. It has short telomeres, but its telomeres shorten very slowly. So it has a really long life. So it's not how long your telomeres are, it's how the rate of shortening that actually affects the lifespan of the organism. So even with, an epigenetic reset, you still need cellular division. And when the caps of your telomeres get critically short, your cells do not divide anymore. And that's when we go into senescence, meaning that drives another hallmark of aging, where cells cannot replicate, and they start to send off inflammatory markers. And this is the point in which almost all of the population starts to become diagnosed with one or more diseases of aging. So the diseases of aging: cardiovascular disease, heart disease, kidney failure, cancer, those are all symptoms of old cells.
Okay. Well, so let me ask about the procedure that you undertook in Colombia back in 2015, because again, for the audience, you actually undertook a procedure to lengthen your telomeres. So I'm wondering if you could tell the audience a little bit about what it was that you did and why you had to go to Colombia for it, I guess.
Yeah, so in 2013, when my son was diagnosed, I was highly committed to being part of the change, to change the sick care system into a healthcare system. BioViva was born out of that. First, I started by creating an industry, or trying to create an industry, around funding these types of technology. Everyone that I ran into said that the stopgap in longevity was money. But I didn't have, strong enough connections to really make that work. I traveled all over on my own dime, trying to get investors to invest in the technology. And what I heard over and over again is, if the technology worked, we would fund it. And I would show data of how it did work in mice, and how this technology had been around for quite a long time. The two therapies that I fell in love with were telomerase reverse transcriptase, the telomere lengthening gene therapy, and another one called follistatin that increases muscle mass. And we decided in, I think, 2014, that we would start a company that would actually prove whether technologies worked or not. And I didn't know how disruptive we were being, even to our own industry. We said, if you have a therapy and you think it works, and 41 million people are dying every year, then let's take it. And that's not even how biotech apparently usually works. It's more about raising money around an idea. And we thought, why raise money unless the technology works? So anyway, we still challenge companies to come up and do that with us. And so in 2015, I left the country to do 2 gene therapies. And the reason I did that is we did not want to upset the US FDA. We wanted to do everything as legally as possible as a company for the success of our own company. But we felt that there was a mandate to try these 2 gene therapies in a human, because these could actually save billions of lives potentially. And with the follistatin, the muscle enhancing gene therapy, had already been through safety and efficacy in the United States for Becker's muscular dystrophy with Nationwide Children's Hospital. So we felt really pretty confident about that. And actually, the doctor who gave me the dual gene therapy had already done follistatin himself. for performance reasons and knew it worked. So the telomerase reverse transcriptase was brand new. People speculated that it would cause cancer, whatnot, but we went out of the country and did the therapy to, break new ground and say, this is it. We either have it or we don't. And since my view was really on children, I mean, I knew we were taking a big risk at the time because it hadn't been used, but I don't think that I could have lived with not doing anything. I mean, seeing my son struggle with disease, and every trip to Children's Hospital is, kids who are going through cancer therapy. And if you're really unlucky when you're there, they have one of those codes come over the intercom of which a child is dying. It's incredibly sad. When your kid is sick, you don't live in this world of like, everything's hunky dory and someday I might get sick and it doesn't matter. I mean, you're dealing with illness every day. And that really kept us abreast of wanting to be part of that test. And I certainly wanted to be part of that. And as a company, it really wasn't fair for us to choose some hapless person who might not understand the technology. So I put myself up for that.
Okay. Well, I understand, I've read that your telomeres were considered short before the procedure. Hopefully I'm not mistaken on that, but
no. And I can show that. I have my tests here.
And so after the procedure, there was measurable lengthening. I'm wondering, can you tell me a little bit about, I guess, what was measured and what that means?
Yeah, so that's the important thing, is really what it means. And so when they measured telomeres with the tests that we did, and we've used 3 different labs to verify across the board on different tests, the length of my telomeres, they only test the T lymphocytes. And that's kind of a problem because telomerase reverse transcriptase, the enzyme telomerase, is not shared between cells. It's only viable in the cell in which you transfect. And we know that we can't target all the cells in the body yet with gene therapy. And that's why one might do a gene therapy more than once, like I did. And so, because that's a limiting factor, we can only go with the tests that are available, right? So we know that in the lung, the brain, and different places in the body, the telomere lengths might measure differently. But again, this is what was available at the time. And so when we do a test pre-treatment, we're going to continue to do that test post-treatment. So what we can see in my graphs is lengthening of my telomeres and continually lengthening of my telomeres over time.
Okay. So in terms of the procedure itself, you've talked about after the procedure, I think you've talked about sleeping better at night. I'm wondering, were there any other positive effects? And over the years that have passed since this experimental treatment, have you seen any other long-term positive effects?
Yeah. So like I said, I've got my data here and I'm happy to show it and walk you through it. But as far as like the testimonial side of like the, anecdotal things of like what I feel and see, I think that really being in a better state of health is interesting because it's not really what you feel, it's what you don't feel anymore. So, if there's something up high, I can climb on the countertops and get it. I can, lift more weight. I tend to not suffer from illnesses the way that people around me do. I sleep better. Like being in a state of health is kind of like the absence of pain and problems and limitations. And so that is really the great part about taking part in the therapy. We work with a group called Integrative Health Systems to assess their data. And this is something that we are hearing over and over again. Certainly another side effect, and we showed this in mice in our work with Rutgers with these 2 genes, hair. like hair. My hair has not been this thick and this long since I was in my 20s. I mean, it's an interesting side effect, but it's been proven also in animal, in organisms, model organisms, that is the case with both follistatin and telomerase reverse transcriptase. It's not a cure for everything, obviously, and we haven't cured aging, but this is the beginning to doing just that. So we're really excited about being part of that.
Yeah. Well, now, have there been any negative side effects?
No, when you're, that was something that we looked out for a lot. We didn't know what was going to happen. I kept a journal when I came home, and going back and reading it is really kind of shocking, because, there was worry. There was like, I don't feel good today. I'm not sure what that is. I didn't tell my family I'd taken the gene therapy. I'm so glad, because I came home and the house was just mellow. They were just expecting me to go do the dishes, get the kids something to eat. And that sort of normalcy sort of like kept me sane. I remember right after taking the gene therapy, calling one of our advisors, and he called another advisor, and the advisor gave me like 13 days to live. He's like, I give her 13 days to live. And I was like, now we know, because more studies have been done, that telomerase reverse transcriptase is really everything that it promises. It will probably protect us against cancer. It doesn't cause cancer. My initial treatment, we injected into my breasts, anywhere where I would be exposed to massive amounts of sun, any of the really cancer high risk areas, to see if, it would, I certainly didn't want anyone else to take this therapy if there was a risk at all. And we have seen no cancer, no negative side effects. And this is really the benefit of gene therapy. So when people look at it, they'll say, well, how could it not have a side effect? But that's because they're used to taking old medicine, right? They're used to something like a statin that may reduce their LDL cholesterol, but it also, 1 in 4 people will get type 2 diabetes, 1 in 10 will get dementia. It affects your hormones, it affects a myriad of things. We're just used to bad drugs that were once really good drugs, but the technology now has moved well beyond that. So, any drug that you take that your doctor prescribes is more than 15 years old if it was just regulated. The breakneck speed that science is working at is just, we're so far beyond that. When we look at genes for upregulation of regeneration, we're looking at specifically a gene that does just that. We don't have to choose something that might have a myriad of off-target effects, but that's not to say that some gene candidates won't. These ones had been well studied for, over a decade, and did not in any model organism, and did not in my human body.
Okay. Well, so from what I understand, telomeres, the shortening of the telomeres, is only one component of aging. So I'm wondering, is BioViva exploring other avenues for lifespan extension, or are you guys primarily focused on the telomeres?
Oh, no. Yeah. A lot of people think that we're just focused on the telomeres. And as I told you, with my first gene therapy, I did 2 genes. And so we're definitely not. And since then, I've done 4 genes. So we have expanded our playlist by 2 more genes, and we're still looking at candidates. We're also looking at cell reprogramming, because one of the things that you need to do with that is also lengthen telomeres. So we're doing some studies there. So we also work with a gene called Klotho. Klotho is just an absolute dream gene. It protects you against cardiovascular disease and chronic kidney disease. And it also helps with cognition. So we took part in the biostatistical data of the first gene therapies ever used in dementia patients, in which both telomerase reverse transcriptase and Klotho was used. And it's because they're both associated with less beta amyloid plaques, less tau tangles, but better than that, upon autopsy, people who had essentially had Alzheimer's but didn't suffer the cognitive decline were natural upregulated individuals of the gene Klotho. So it's associated with cognition and better memory. And so I was excited to take part in that gene therapy. And, I mean, it's interesting. It could have been the day, but on my IQ scores, they went up after I did the dementia treatment. Again, I've tried to participate in anything in which other people might be participating in to do my own safety study and to see if those things are actually going to be okay. And another gene that I participated in is called PGC1 alpha. It's a gene that we upregulate when we exercise. So you can go out and run your little heart off and upregulate the gene. It's also a gene that you can take in a gene therapy, and it's just associated with better cellular communication. And so, yeah, our genes keep expanding. And then, of course, we're working with some of the Yamanaka factor genes as well to see what we can do on the programming.
So yeah, there are a lot of different factors. Then it's not just telomeres. It sounds like there's this entire spectrum of genetics that you guys are looking at.
Oh yeah, and we continue to look at genes. We look at the BDNF. We look at all the gene therapy studies going around cardiovascular disease and some of the genes that they're using in patients who have heart attacks that help repair damage more quickly to the heart afterwards. We will actually be releasing the Code Keeper in the next week or two, which is a free place that you could go if you have a disease or you're interested in specific genes, and you can see everything in gene therapy happening in that space. And if you don't find it and you know about a paper, you could submit it, and the PhDs on our side will parse the data and put it into the database. It's going to be a really cool tool for people to use. So if they have a disease or an issue, they can actually go look up what might be available for them. And of course, we use those databases, we use clinical trial data, we look at everything in order to create better therapies for you in the future.
Okay. Now, so one of the things that I definitely want to touch on is you had mentioned the speed of approval for drugs and treatments. And I think you'd said it's probably about 15 years, and that's for something that's brand new, right? And so if it's been out for a couple of years, it's even further out. So I guess I'm wondering, have the laws started to change, and was the experimental treatment that you went through, was that kind of partly making a statement about the approval process?
Well, I mean, for us, we weren't saying anything about regulation at the time. We were just saying that we wanted to respect the laws of the United States. I'm a big, patriot of the United States. I've traveled all over the world. I love this country. I love our infrastructure. Now we have to protect it to make sure that it stays up to date. We have some roads falling apart these days, but, so at that time, it was just, we wanted to play within the rule book of the United States. Since then, I went on to do my MBA. I focused on regulations because it's a huge problem of what's wrong. It's a huge issue as to why people are not getting access to these technologies. And so I did my thesis based around the regulatory system and a new framework in which that's how I concluded. I didn't know that that's how I would conclude. But I will hopefully, I've got some people working on it with me to distill down the thesis and release a paper in which we will try to get published through peer review, and also give to the United States government for consideration of a new route in which would give access of this new technology to patients who are dire need now. It's better than any of the technology before. It's more exact, less side effects, less risk, especially for people who are dying. And, right to try and advanced access options don't give access to this type of technology. And so we really need a regulatory route that does, and, we need to, as a country, stay on top of innovation. Nobody should be leaving this country to innovate. We should be doing it right here. And so, yeah, that's how much I'm a fan.
Yeah, no, that makes sense. Well, Another thing that I wanted to touch on, and this is maybe a little bit more topical, but the COVID pandemic, which is still more or less going on, has cost trillions of dollars. And the goal has been to prevent the spread of a disease with a 5% mortality rate. So in contrast, aging affects 100% of the population and has a 100% mortality rate. Why do you think more resources aren't being put into prevention and ultimately cures for aging?
Well, I think that it starts with our scientific institutes. Well, number one, it would be great if we could get aging considered a disease, because then you can put more funding into solving the problem. I believe it's a disease. It's a pure pathology, and the symptoms of that pathology are dementia, heart disease, cancer, you name it. They're all of the diseases of aging. And so that's why we haven't been able to cure them, because we have been treating the wrong thing. We've been treating the symptoms of a symptom. So if you treat beta amyloid plaques, you're treating the symptom of Alzheimer's, which is the symptom of aging. And that's going to be a problem. You're never going to cure anything that way. But if you get down to the cellular level, what's happening at the cells level in which the degeneration is being driven, the hallmarks of aging, and those agreed upon hallmarks will change over time. More things will be added to them, which gives us more targets to go after. Then you do have the ability to cure disease and extend health span. So a lot of people will say, well, maybe I don't want to live longer. Maybe like 85, 90, 100 would be fine for me. Well, you can choose your death. I don't suggest it, but you can choose when you want to die, maybe. But if you are healthy at those ages, right now we spend a third of our life in ill health. I mean, that means that you and me and a bunch of people our age are just running right up against diagnoses in which, the rest of our life won't be so great. Why wouldn't we expand the health span there? And you're right. This year, 41 million people will die of aging-related disease, most of them prematurely. 74% of the population now dies of non-communicable diseases, and the top 4 non-communicable diseases are aging diseases. This is something in which we need to pour a ton of energy into. But there's a slow shift at the scientific level of what people are working on, and luckily people started working on this decades ago, and so that's why we're coming to the fruition that we are. That's why I was confident enough to take a telomerase reverse transcriptase gene therapy when it hadn't been in a human, because there was a lot of data that showed that it would probably be very safe and probably the best drug on the planet for a baseline of getting people healthier and protecting them against things like cancer. So things shift slowly. There's a lot of competition in science. Scientists often don't speak very well of each other. People in the industry often hurt other people in the industry by making wild guesses and just saying things that aren't scientifically proven. People have come to me and they've said, I was told that telomerase reverse transcriptase would not cure aging. Well, by who? Because a proper study has never been done to null that hypothesis, but it could be possibly true, but it still is a key ingredient. So we don't, there's a lot of things that are said that are just not based in science that make investors go, oh, well, that one gene might not cure all of aging. Well, there's probably not one gene that would. And that's why we're doing the combinatorial gene therapies at Rutgers now. So in human cells, we're actually combining genes and seeing if we can get a bigger bang for the buck for a larger dose candidate of genes. But all of the technology needs to be funded. No one needs to be talking bad about each other, because we need it all funded, because everybody's working on a different part of treating aging. It's not a zero-sum game. And so, I think that mostly we need to make sure that we're not hurting each other whilst moving forward, falling in love with our own hypothesis. And I try so hard to not fall in love with my hypothesis. That's why we look at a wide variety of genes.
Yeah. Well, so I guess one of the things that I really wanted to kind of touch on is because you've had more time to ponder this and consider this than other people, I guess. But as human beings, I think culturally and personally, we have this view of the inevitability of aging. And so what I'm kind of wondering is, do you think when that starts to shift, and I think it's already starting to, but as that starts to shift, do you think that will change the amount of funding, the amount of interest, the amount of push towards treatments like this?
Oh, yeah, absolutely. I think that we have to envision a different future. And, we have to stop starting at the wrong part of the conversation. For instance, people go right to, oh my God, if people live longer, what are all the terrible things that will happen if people live longer? Well, we already live longer. On average, we're living 3 times as long as we used to through science and technology. And we're continuing to solve problems. My company doesn't solve certain problems that need to be solved for the future, which is, good, clean energy and, protecting our planet and things like that. But people are working on that, and I trust them to do an excellent job there. So instead of starting at what happens if people live longer, oh my gosh, we should start at, as soon as we can cure dementia, we should do that, because it's inhumane. What we should be doing is ensuring that our focus, we should be asking critical questions, but we should make sure that our focus is on a better world, not on the worry of things that, of course, we'll take care of as we go. The world is becoming a better place. If you look at our world and data, we're becoming a better place all the time through technology. Hans Rosling wrote a great book about it, where a lot of people, especially people in the media, don't understand or see the improvements and how many people have been lifted out of poverty and how lifespan has extended around the globe, which is a sign of good science. They don't know that, and it's an education in itself. We live better through the technology that was made by our forebears, and then we will continue to do that. And I think that people will fall into sort of a timeless span. People already look younger at older ages because of better living, and they will continue to do so. And eventually it will blur the lines of, I imagine that in the future, the older you are, the more excited you'll be to talk about it.
Yeah, absolutely. Well, so Liz, let me close by asking, what comes next for you personally, and what do you see in the future for BioViva?
Well, personally, what comes next for me is, I'll probably be designing whatever next gene therapy I'll be participating in. That's exciting. We'll be releasing our paper about what happened over all of these years with my gene therapy. And we are combining genes now to see how they perform in human cells. We're working with a group outside of the country, potentially to do a non-human primate study that will help us get into phase 1 trials in the United States. We'll continue to push for a larger cohort study and bring in donations for that for dementia. We'd like to do a larger study with 20 people and see if we can get the same results that we did with the first 5-person study. And, we work every day. We work 7 days a week on, preparing the future for humanity, for a better future in which we suffer less. So that's pretty much it.
Wonderful. Well, thank you again. And actually, you had mentioned having some graphs and charts and data. If you'd like to share those, go for it. If you want to do a screen share or anything like that. Yeah,
Sure.
I'm still recording, but we can, I can put this up.
Okay, so
yeah, I would love to have that up, and then I can add.
Can you see this screen?
Yeah, it looks great.
Okay. All right, so we'll just go over this really quick, because this is the stuff that's been accumulated for the paper that'll be coming out. So you'll get to see in advance some of the graphs and stuff. So this was my before. We've got a new one coming. I just did an MRI on my thigh, well, just last Friday. last Friday evening. And so we'll have a new MRI coming out, and we have years worth of MRIs, but this was the before, and this is the effect of follistatin. You can see this is my before. This is when I was running about 30 some miles a week. And this was my after, when I had stopped running and I was traveling with the company, and I stopped to go take a MRI. And you can see that I have less intermuscular fat, which is great. And I have more muscle mass, and it's denser. And so that's something indicative of follistatin. I mean, it's really like, it's a great gene therapy. And we've learned a lot about that over the time. Oh, I think this is just the summary of all of my blood work. The problem with the blood work is we can see some really great things here, especially in HbA1cs and in blood glucose. But these will be a supplement to the paper. They won't be part of the paper, because doctors consider these sort of readings to be transient. And so it depends on what you eat before, or if you didn't eat or whatever, even though you do most of them fasting. Anyway, for the doctors that worked on it, they just said, yes, your trends are so much better and everything like that, but blood work comes and goes. And so it was not as interesting to them, although I think that some people could totally nerd out with the analysis of those blood work data. This was my telomere length. So this is, I'll show you graphs, but these were all of my tests. This was the before, and no, sorry. This one is the after. Let me see. These may not be in order. Hang on. I may have to go to, okay, that's the before. Okay, so this was the before. And so, you know, this was my average age, was really in about the age of a 65-year-old. This was right after the gene therapy. Oh no, this was some months after the gene therapy, and then they're right on my age. And then we can see here, we start to get lengthening. We can continue to get lengthening, and we continue to get lengthening. And then that's kind of hard to understand what you're seeing there. So that was all put into a biostatistical graph that talked about the first gene therapy and then the second gene therapy here. And this is what we saw over time. So this is my average telomere length since pre-treatment of my first treatment and through the second one. And so the average telomere length increased by 0.35 kilobases per year. And that's actually really good. You know, they didn't get extremely long really fast. They just seemed to be kind of tooling along and lengthening over time, which is what we would expect with a long-term gene therapy. And then this one is, compared to the patient population. So my rate of telomere increase was 9.1% per year. And so that was really fantastic. Associated age over time went down. So I gained in my telomere length 5.3 years per year. So in my telomere length and my T lymphocytes, you know, you have to be clear about what that actually means. These are just my T lymphocytes. So this was really great. I mean, this is, you know, beyond how I feel. This is the actual data. So this is what happens when you take a gene therapy. So, and that, again, these things will be in the paper that will come out about the test itself.
Wonderful, wonderful. Well, Liz, thank you again. Thank you so much for your time.
You're welcome. Thanks for having me.